Okay, I guess we'll get started. My name is Keith Berven, as you probably know, and I'm going to give a presentation about soils. And since we have limited time and I have a fair amount of information, I'm going to try to go through it quickly, and hopefully there'll be enough time afterwards for questions, but if not, feel free to track me down and ask your questions. Many of you, probably like me, when you started growing dahlias, somebody told you you should get your soil tested, which I did, and then I got all of this information and had no idea what any of it meant. And so that started a process of trying to learn as much as I could about soil and what these various parameters were measuring. And that's what I'm going to present to you today. You should realize that I am not a soil scientist. I'm a biologist, particularly ecologist and evolutionary biologist, but because of my interest in dahlias, I've spent a lot of time worrying about my soil. So here's what a typical soil test looks like, and I've just highlighted some of these parameters that we're going to be talking about today. And what I thought I would do in this presentation is to give you a little bit of background about some of the characteristics and principles of soil science, and then actually go through each of the parameters that you will receive when you do a soil test. So the first thing is going to a little bit about soil structure. So if you look at soil structure, it consists of really four things. These are the minerals, the soil organic matter, air, water, and the microorganisms, the bacteria and fungi that are present in the soil. If you look at the mineral component of soil, it's primarily sand, silt, and clay. And these differ primarily in the size of the particles. So the sand is large, it's something you can see, you're familiar with it. Clay, on the other hand, the clay particles are microscopic, so they're very tiny. And all soils are usually made up of a mixture of these three different particles. And as you will see later, these are very important in terms of maintaining and holding on to the nutrients and minerals in the soil. Once you get the amount of silt, sand, and clay in your soil, the USDA has classified soils based on the percentage of each of those categories in your soil. And you can see with this diagram, you can draw arrows, and where they all intersect is going to be the type of soil that you have. And we'll talk more about that in a minute. But the soil texture, the sand, silt, and clay, is very important in a variety of different ways. As I already mentioned, it helps hold on to the nutrients in your soil. It participates in soil drainage and also water retention. It affects air movement in the soil, and which is related to root penetration and respiration by the roots, and then also the microbial activity in your soil. So just to give you two quick extreme examples, if you have an extremely sandy soil, that means it's generally well aerated because there's lots of spaces between these large particles. And that's very good for aeration, it's very good for water flow. The problem is, one of the big drawbacks of sandy soils is that it reduces the amount of water available to the roots because it percolates through very quickly. As if you have sandy soils, you probably realize you have to water a lot more frequently. And also, because sand has a very small, low ability to hang on to nutrients, and there's a lot of water flow through the soil, it tends to leach all of those minerals out. So sandy soils on one end have a lot of advantages and disadvantages, whereas silt and clay soils have a benefit of being able to retain water and retain minerals, but those can also be drawbacks because they'll tend to retain water, which can lead to problems of rotting. And because the soil is so well packed, it's very poorly aerated, so a lot of air isn't able to get to the roots, and the roots have difficulty penetrating the soil. So in a nutshell, having a balanced amount of sand, silt, and clay, and we'll talk about what that balance is in a minute, is basically what's going to provide you with the the best soil. The second thing that soils are made up is soil organic matter. Soil organic matter is basically three major components. There's the soil biomass, which represents all of the fungi and bacteria and other living organisms that are in the soil. It's the partially decomposed plant and animal material like leaves and roots and animal waste. And then it has humus. Humus is the final end product of decomposition by microbes and fungi in the soil. And humus is what gives soil that dark brown color, and generally the darker the soil, the more humus that's present. But all three of those things are part of soil organic matter. And the importance of soil organic matter cannot really be overstated. Those leaves, twigs, all of that raw organic matter basically houses a variety of important minerals that the microbes are going to break down. This process is called mineralization, and they will release those important nutrients to the soil for the plants to pick up. Soil organic matter and humus in particular, can hold 10 times more water than a mineral soil. So the more humus, soil organic matter in your soil, the better ability to hang on to the water. Humus combines with clay particles to kind of make the soil more crumbly. It improves soil tilth, it improves aeration, it improves water flow, and it improves root penetration and reduces soil compaction. And humus, as you will see, also has an extremely high, actually 50 times higher than clay particles, in what is known as cation exchange capacity, the ability to hang on to these minerals. And we'll talk more about that in a minute. So soil organic matter is something that you should always pay attention to in your dahlia gardens. Now, soils are generally categorized as either being mineral or organic. Minerals are generally the case if 20 to 30% is organic content, organic matter, soil organic matter. And if it's over 20 to 35%, the soil is usually described as being organic. So oftentimes you'll see in your soil report that your soil is either mineral or organic, and this is what they're referring to. Obviously, ways you can improve your soil structure is by adding organic matter, leaves, straw, any kind of organic matter. Encourage earthworms, add mulch, limestone. The things you can do that we invariably do, but maybe not realizing, is you can destroy the soil structure through walking around on it all the time and compacting it. It's always important to have areas to walk where you're not walking directly on the roots of your dahlias. And also, if you cultivate the soil when it's either too wet or too dry, you'll completely destroy that structure, which leads to the good aeration of your soil. Air is another component of the soil structure, and it sort of goes without saying that oxygen is required for respiration, and that respiration, the oxygen, is used by roots. And so in order to have a good aerated soil, that will allow the oxygen that's needed for cellular respiration. The same with water. Water turns out to be a compound that is a source for hydrogens and oxygens in many of the carbon compounds that plants produce. Many minerals, as we'll see, are dissolved in water and taken up that way through plants. And you probably are aware of how different your plants are during the course of a sunny day in terms of their rigidity. When they're turgor pressure, when they're full of water, basically they're very stiff, and in the middle of the day in the hot sun, they lose a lot of that water and they get very limp. 90% of the weight of a living plant is actually made up of water. So obviously plants require water in order to grow. And then finally, the last component of a soil is the microorganisms. So you've probably heard, if you take a teaspoon of soil, there's probably roughly 55 billion bacteria that cohabit with a variety of other soil microbes, such as fungi, protists, and other organisms. There are larger multicellular organisms like insects and worms and nematodes, and even the roots of plants, as you'll see, are somewhat involved in the, some of the characteristics associated with the soil. And so many activities of these animals help basically maintain the proper soil structure. So earthworms, through their burrowing and through their depositing of mucus, tend to help aerate soils, and they also tend to hold fine particles together. The bacteria, that process of mineralization, the bacteria's process of basically eating and decomposing that organic matter, basically releases minerals like phosphorus, calcium, into the soil so that your plants can take them up. And the roots also are extracting waters and minerals and can also affect the soil pH and other characteristics of the soil. So microorganisms are a big part of your soil chemistry, and they're a very big part of the soil organic matter. Now, one of the aspects of the microorganisms that you've probably heard about, the mycorrhizal fungi. So these are fungi whose mycelia, which are basically the roots of the fungus, that little toadstool that you see is actually the flowering part of the plant. The rest of it is underground, but mycorrhizal fungi are beneficial fungi. They form relationships with the roots of plants, and almost 90% of all seed plants have specific relationships between these mycorrhizal fungi and the roots. Some actually penetrate the cells of the roots, some just surround the roots. But what they do is to basically have a symbiotic, which is there's a plus-plus relationship. The plant benefits, the fungi benefits, in terms of what each of them are getting. The fungi obtain organic food from the plant, and the plant supplies a variety of different minerals and water. And this becomes a very efficient and important way for plants to be able to sample a much larger area that they're growing in than just where their roots are. So just give you a quick example. Phosphorus. We all know how important phosphorus is, particularly to flowering. And so we all want the best flowers that we can have. As you will see, most of our soils have way too much phosphorus in them, but that phosphorus is not available necessarily to the plants. But the fungal associations dramatically improve the ability of the plants to pick up that phosphorus. And here's an example. So if you look at that diagram on the left, you can see that big finger-like looking root, plant root, and then you can see under that a little green line. Well, the soil immediately next to the roots is going to basically absorb all the nutrients that are available in that soil. And once those nutrients are gone, those roots obviously are not going to be able to absorb any more nutrients. Here you can see the mycorrhizal fungi, which are associated with those roots, now extend way down into that brown area. And on the right, you can see the mycorrhizal hyphae, the roots of the fungi, extends much farther. So that means they're able to sample the soil not only for nutrients, but also water. And so there's a huge benefit for the plants. And so this is something, again, that we need to try to encourage in our gardens. A lot of things affect the mycorrhizal fungi, but the factors that tend to negatively affect them, tilling. Tilling is generally something you're going to basically disturb the structure of the soil. All of those fungal mycelia are going to basically be broken up and disturbed. While pesticides applied to the foliage don't seem to have an effect on the mycorrhizal fungi, if you put any kind of fungicide in the soil, a fumigant, these will definitely get rid of not only the bad fungi, but all of the good ones. So it's something you should consider. So mycorrhizal fungi are an integral part of the soil. All right, what about plant nutrients? There are two types of plant nutrients, the macro and the micro. You can think of the dahlias out on the table there. We've got the macros, and then we have that table of little tiny micros. Basically, the distinction here is that the macronutrients are needed in large quantities, whereas micronutrients are necessary, but only in very small quantities. And so anytime any of these nutrients is missing, it's going to have a negative effect on the plant's growth. And here are what are known as the plant macronutrients. So in addition to carbon and oxygen and hydrogen, we have things like nitrogen, potassium, calcium, magnesium, phosphorus, sulfate. These are the things that you're probably familiar with that you see are present in your fertilizers. These are the things that plants need in relatively large quantity. But what I want to point out is this column here that says percent dry mass in dry tissue. You can see that once you get down to something like nitrogen, which is at 15%, so 15% of the dry weight of a plant is nitrogen, then we get down into the neighborhood of 1% or even less. But that's still, these are still macronutrients, because if you look at the micronutrients, you can see that the levels that are required there are orders of magnitude lower. But these are essential, and they must be available. And these are things you probably have also seen in fertilizers, such as manganese, boron, zinc, molybdenum, which is another one. But these are the micronutrients. Both are needed. Both are needed in drastically different levels. Now, these macro and micronutrients occur in two forms. And the reason for bringing this up is you'll see that it relates to some of the other aspects of the soil parameters that are going to be measured. There are some that are positively charged, and these are called cations. And you heard me say cation exchange capacity. I'm specifically referring to the cations such as potassium, calcium, magnesium, and sodium. These all occur in the soil with a plus charge. Negatively charged nutrients are called anions, and these are things like nitrate. This is the form of nitrogen that plants pick up, phosphate and sulfate. So these are anions as opposed to cations. So these are the two forms that they come in. This brings us to cation exchange capacity. So if you look at those mineral particles in the soil, so we talked about clay, silt, and sand, and we talked about humus. Those clay particles, as tiny as they are surrounded by negative charges. And those negative charges attract and hold those positively charged minerals, like calcium and magnesium and potassium. So that's what's holding on to those minerals in the soil. It's electrostatic attraction. Humus has both positive and negative charges, so it can hold on to not only cations, but also anions. When you see the cation exchange capacity listed on your soil test, what it's doing is measuring the total negative charges in your soil, which is an indication of what is the maximum number of cations, positively charged minerals, that your soil can hold, and that's what that is a measure of. Clay and humus, as a consequence of being negatively and positively charged, basically are the parts of your soil that help reduce leaching of minerals during a heavy rainstorm, or if you're irrigating and there's a lot of water flow through your soil. And these small little particles, for their size, have a lot of surface area. And that surface area is where all those positive and negative charges are, which are attracting those minerals. And so the cation exchange capacity can be used to basically predict the nutrient availability in your soil. The higher the number, the more likely there are more nutrients available, and the better ability of your soil to retain those nutrients during watering, for example, or rainfall. All right, Many of you probably often wondered how plants take up nutrients. You think, well, the roots just grab them and pick them up. But the roots are actually involved in something that is rather interesting. The roots are releasing, as you can see in this diagram on the right, water and carbon dioxide. That carbon dioxide combines with water to form an acid, which then dissociates, releasing hydrogen protons, so those little H pluses. So those are positively charged structures. Those positively charged structures go over to those little humus and clay particles and displace the positively charged minerals, making those minerals available to the plant root to take up. And so the roots are actively involved in this chemical activity, changing the pH of the soil in order to be able to absorb these nutrients. Negatively charged ions, so these are things like nitrate, for example, they're absorbed by the roots in a slightly different process. The negatively charged particles are not held by the clay particles, and instead they remain dissolved in the water, the soil solution, and they can move into the plant roots in two different ways. One is by mass flow, so as the roots are absorbing water, and usually this is a process that occurs as the roots are losing water through evaporation through their leaves, it tends to pull water out of the ground and into the plant. And water is what's called a polar molecule. It has plus sides and negative sides, and they kind of cling to each other. If you put water in a small tube, you see a little edge that comes up, which is called the meniscus, and this is basically the result of that polar activity of water. So as water evaporates through the leaves, it's pulling it up from the soil into the roots, and it's basically connected all the way from the roots all the way to the leaves. As that water gets pulled in, any nutrients that are dissolved in that water get carried along into the plant as well. The other way that it happens is by simple diffusion. If the nutrient is more highly concentrated in the soil than it is in the roots, it will diffuse down a concentration gradient from where it's more highly concentrated to where it's a lower concentration. So those are the way the negative ions the anions basically are absorbed by plants. And we'll come back and talk more about that in a minute. All right, so with that brief background, I now want to basically go through and talk about the parameters that come in a typical soil science test and what they mean, what are the desired levels, and what are optimal for dahlias. So here we basically have a description of those parameters. So the first thing you usually get is a description of your soil. So this is going to be whether it's mineral or organic, based on looking at the percentage of organic matter, the soil, sand, silt, and clay. It'll also measure the organic matter in your soil, and those two things together what they will use to describe in that triangle of your soils classifications what type of soil you have. They will also measure soil pH. They'll measure the cation exchange capacity. Remember the total negative charges that are present in your soil. Another one related to the cation exchange capacity is something called percent exchangeable bases. And so this is specifically referring to 3 and a few more plant minerals like potassium, magnesium, and calcium. And then the last part of a typical soil test is to tell you how much phosphorus, potassium, and magnesium. So these, if you're used to looking at fertilizers, the NPK, this is that ratio that you're looking at. These are the ones that are obviously macronutrients. They're important for good plant growth, and so they will tell you the levels of those in your soil. So let's go through and look at each of these. Basically, depending on the company doing the test, they will give you some information or just tell you. But here's that diagram again for the classification of soils. The ideal dahlia soil is referred to as a sandy loam. And you can see the sandy loam is down there in that purple area. And this basically represents about 50 to 60% sand, 0 to 10% silt, 10 to 25% clay, and 6 to 25% organic matter. So within that purple area, that basically satisfies that range of silt, sand, and clay and organic matter. And that is considered to be the optimum soil, particularly for dahlias, because there's enough fine particles to produce a large surface area to cling on and retain all of those minerals in your soil. So it leads to a high cation exchange capacity. It also has enough coarse particles to keep the soil loose and the tilth good. So it's well aerated and there's good water flow. And so having a good mixture of all of these things is highly beneficial, particularly for growing dahlias. So if you get your soil test back and it says sandy loam, you're in good shape. If you get it back and it's mineral and it's primarily clay, then you know you've got some work to do. All right, so here is a typical soil test, and you can see highlighted there, it shows you the soil type, and it says mineral sandy loam. So mineral in the sense that it doesn't have an overly high amount of organic compounds, but basically it's a sandy loam because it falls down in that category of having a certain amount of sand, silt, and clay, and organic matter. And some tests will go as far as to give you the actual breakdown of the amount of clay, silt, and sand, so you get an idea of where those numbers are coming from. If you look at a variety of different dahlia soils gardens, so I'm going to be showing you a lot of summary soil tests for a lot of different dahlia growers. These are sort of the typical descriptions of their soils that they get. So many are sandy loam, many are mineral. It's pretty typical for most people that are growing dahlias. This is the type of soil that they generally have. How about organic matter? This is the other measure that we're going to come out of your soil test. So here you can see on the bottom, it gives you a percent organic matter in your soil. And this one says 6.5%. Organic poor soils, so the soil organic matter in a soil is very bad. It's usually going to be less than 1%. And anything over 8% is generally considered very good. And as you'll see, most of us fall into that category. Optimal growth, basically the optimal soil, is usually going to have something greater than 8% or somewhere around 8% organic matter. And again, this is related to the presence of the clay particles and the humus particles that have that ability to hold on and retain the minerals and make them available to the plant. So here are the pHs of a variety of different dahlia growers. And so generally, 8 to 16% is, but really the sky is the limit. It really, there's nothing wrong with having too much organic matter. And so you can see that most dahlia growers basically are maintaining fairly high levels of organic matter in their soils. All right, there's an interesting thing you can do with those two pieces of information, the percent organic matter in your soil and the cation exchange capacity, the ability of your soil to hold on to those minerals. Remember, one of the particles that hangs on to those minerals are the humus particles. Remember, humus is the final breakdown of soil organic matter. So if you look at the relationship between the cation exchange capacity of your soil, sort of the sum total of all the negative charges holding on to those minerals, and here we're specifically looking at the humus particles, and you look at the relationship to the cation exchange capacity, you can get a little bit of an insight into how the biological component, all the fungi and microbes in your garden are doing in terms of the population size and their ability to break down and mineralize that organic matter. So here is a list of the pHs, the organic matters of different dahlia growers' soils. And then in parentheses, I've put the cation exchange capacity in white in the middle of each of those bars. And so the general theory is that if you have soil organic matter that's greater than 5%, which is good, but you have a cation exchange capacity that's less than 20, what that means is the biota in your soil is not healthy. It's not doing the job that it should be doing, and you need to promote greater biological activity to increase the rate of decomposition and turning that material into humus. And you can see that basically many dahlia growers fall in the appropriate range, but there are some dahlia growers that need to encourage the soil, the microbes and fungi in their soils. Just to give you a little from my own garden, when I saw this and got interested in it, I thought, okay, well, I've been taking soil samples for a long time. What is the relationship between the percent organic matter in my soil and the cation exchange capacity? So the cation exchange capacity is in purple, the percent organic matter is in green, and you can see that statistically there's a very strong positive correlation between those. So encouraging the biological function, addition of organic matter, compost, basically leads to an increase in the cation exchange capacity of your soils. Now, how do you improve the humus content in your soil? Obviously, you need to add organic matter. Compost is the main thing that most people add, but you can add leaf litter directly, manure, cover crops such as legumes and grasses. These materials contain high levels of organic carbon and eventually will be broken down by the soil microbes. And this is particularly true of mulch, which has already had that process going on for a while. Reducing tillage. Again, reducing the tillage tends to disrupt all of these microbial communities that are living in your soil. One of the things that I have found, and what I used to do is in the spring, I'd have all of this, the cover that I had on top, straw on top of my dahlia beds, and I would turn it all over. I thought this is a good thing to get it into the ground and get it broken down and worked on. And what I noticed was that resulted in a huge decrease in the amount of nitrogen in my soils right when I'm planting and when those dahlias need all of that nitrogen. What was happening is adding all of that organic matter to the soil was feeding all of those microbes. And the microbes used up all of that nitrogen to break down that organic matter. And so by now just leaving it on the surface, running over it with a lawnmower, I'm surprised, but after a month, it basically just disappears. It basically gets pulled down into the soil through the actions of earthworms and all of the microbes, and it disappears, and you don't have that sudden spikiness of increases and decreases in nitrogen. There are commercial ways that you can add humus to your soil. Bicar is an example of one. There are different ways of doing it. But the best way to do it is, I think, by adding a good high-quality mulch that has had lots of good organic matter and has a good biology component to it. All right, I don't have time to talk about this one, and it's relatively new in terms of being available. One of the things that those of you that know Tony Evangelista, he hated soil tests because the soil test did not address any of the organic nature of soils. Well, it turns out that now there is a soil test that specifically addresses the microbial communities in your soil. and it looks at a variety of different characteristics. Many of these characteristics are the product of mineralization, microbial decay. It also looks at, for example, the amount of CO2 produced in a 24-hour period of your soil sample. So imagine you need to take in oxygen and then you respire and get rid of carbon dioxide. So they take that soil sample and measure the amount of carbon dioxide produced by those metabolizing fungi and bacteria, and the higher that number, the better the soil biota is. And so this is a test that I highly recommend to see how well you're doing in terms of the populations of microbes in your soil. And it's called the H3 soil extractant test, or the Haney test. All right, soil pH. Again, on the soil test, you're going to see a number, and that's going to be the soil pH. If you're not familiar with pH, it ranges on a scale of 0 to 10, but that scale is a log scale. So what that means is the difference between a pH of 5 and 6 is a factor of 10. It's not a factor of 1. The difference between 5 and 7 is a factor of 100, not a factor of 2. So small changes in pH actually are much bigger than you would think on a strictly numerical scale. pH is extremely important in the soil for a variety of reasons, and I'll just quickly mention those. One of them has to do with the solubility of those nutrients in your soil and the ability of your soil to take up those nutrients. So in extremely acidic soils, a very low pH, this is usually generally less than a pH of 6.5, nutrients like aluminum and manganese, so these are micronutrients that are needed in very small amounts, these become more soluble in that water component, and when roots are absorbing water through mass flow or through diffusion, roots are going to take up these nutrients in large amounts because they're more readily available in highly acidic soil. And as a consequence, in large amounts, these can be detrimental to your plant. In alkaline soils, usually generally greater than a 7.3 or 4 or 5, some nutrients, again, many of these are the micronutrients, become less soluble. So the plant is not able to take them out, and they become a deficiency in your plant. The nutrient exchange capacity. Remember that ability of plant roots to grab on to the nutrients that are on those clay particles and humus particles. Basically, the soil pH can influence the composition of minerals on those clay and humus particles. In highly acidic soils, and what pH means is potential hydrogen, so if there's a lot more potential hydrogen available in acidic soils, those negative charges on the clay particle become more hydrogen particles and less minerals. And so that means there's less mineral availability to your plants. Microbial activity. The ability of microbes to grow, divide, reproduce, basically is affected by pH. So a pH of between 6 and 7.5 is ideal for those bacteria. If the soil becomes too acidic, basically the bacteria are unable to mineralize all of those components. Plant root function. Remember, the roots are releasing compounds that change the acidity that allow the dissociation of those minerals from the clay particles and allows the roots to absorb them. If the acidity changes and becomes too acidic or too basic, the root's ability to use that mechanism to absorb nutrients is interfered with. And it can affect chemical reactions. In an extremely acidic soil, beneficial nutrients like phosphorus can be combined with other minerals and make them unavailable to the plant. So pH is something that you need to pay attention to, and it's a critical component, but I want to stress it's not something to get overly excited about if you see some numbers, and I'll show you that's why in a minute. If you look at this graph, these are basically listed from top to bottom are these different nutrients, primarily the macronutrients at top, the micronutrients on the bottom. Green means that this is the range of pH where those nutrients are best and most easily absorbed by the plant. And you can see for the vast majority of those macro and micronutrients, a pH between 6.5 and 7.5 is pretty much in the green area. There are some micronutrients that basically, if the pH gets a little too high, it interferes with those, but generally you can see there's a fairly broad range. And the reason I bring this up is usually what you'll see when you look at any kind of text on pH in the optimum soil, it'll say 6.4 to 6.8. But I think it's actually quite broader than that. And here you can see the ideal range of the pH in a variety of different dahlia growers. And you can see that a variety of growers have pHs that are much, much higher. And I can basically tell you, based on the results of the show today, that some of these people that have high pHs had excellent blooms that did very well in the show. So you want to try to keep the pH from getting too much higher than 7.5, but otherwise, you know, I wouldn't get too excited about it if it seems way above, you know, an ideal of 6.8. Cation exchange capacity. Again, it gives you a number, and it's measured in this strange number, but remember, it's just measuring the total number of negative ions in your soil. So it's a measure of your ability to hold soil. And so it's a measure of the total negative charges in your soil. And clay and organic matter have the highest cation exchange capacity. Sandy soils have the lowest. Typically, the numbers of the cation exchange capacity are going to vary from 5 to 40. And if you look at the typical range in the soils of dahlia growers, you can see that basically they fall in a range of roughly 12 to 25, which is generally very good. So that, you know, seems to be a very common number for a variety of dahlia growers. Really, there's no upper limit. The higher, the better. The more it's going to be able to hold nutrients and make those nutrients available to the soil. Oh, here's the other one that is kind of a strange thing. So we're looking at those clay particles again. We're looking at all those negative charges on there. But now what we're going to do is say those positively charged cations that are attached to those clay particles ideally should be in a specific ratio. So it should be roughly 65% calcium cations attached to that soil particle, 15% magnesium, 4% potassium. Most soil tests are going to look at those three as the most important, but also sodium is another positively charged cation that is clinging to those clay particles. Hydrogen, remember, those H pluses are clinging to those negative particles. And then all the other bases, which account for about 5%, those are primarily many of those micronutrients. So it turns out from studies that were done about 60 years ago, it turns out that if the soil particles like clay have that ratio of calcium and magnesium and potassium, it is the absolute ideal conditions for plant growth. And I've never seen an explanation for why that's true, but it's true. And so that's the ideal that you're going after. And you can see that it's a zero-sum game. You basically have 100%, and majority is going to be calcium followed by magnesium and potassium, and then you have the sodium, hydrogens, and then the other exchangeable bases. The interesting thing about this is that if the soil has that specific proportion of each of those minerals, the pH stabilizes at 6.4, which I think is the other idea as to why that has often become the magic pH. But remember, what we're talking about are the humus particles, and particularly the clay particles that are clinging to those different nutrients. So your soil test is usually going to give you either a complete or a partial measure of the percentage of each of those cations that are represented in your soil. And so here you can see the typical Dahlia garden soil, with the column on the right being the recommended, and you can see that, more or less most Dahlia growers, the percent exchangeable basis falls in with that range. So what happens if it's not in that range? What happens if you have a whole bunch of calcium, it's 90%? Because it's a zero-sum game, that means there's going to be less potassium or magnesium. It has to be. They're occupied by calcium instead of those. What that means is it's going to be more difficult for your plant to pick up the magnesium and the potassium. But if that ratio is maintained, then your plant has the ability to take those up. All right, finally, the last thing that soil tests tend to report are the levels of phosphorus, potassium, magnesium, and they also usually include calcium. But right now we're going to focus on the phosphorus, potassium, and magnesium. And usually they'll give you some numbers, and there are a couple, usually there's one or two numbers depending on the method they use to measure it. And what they'll tell you is whether it's below optimum, optimum, or above optimum. In an ideal world, we know phosphorus is important, particularly in the formation of flowers and fruits, is something dahlia growers are interested in. Also, it's very important in helping withstand stress. It tends to build up in the soil. Phosphate is very important in plant structure, particularly the stems and stalks. It also is important in fighting disease. It also tends to build up in the soil. Magnesium is part of the chlorophyll molecule, so it's essential for proper photosynthesis in the leaves of your plants. Generally, if you look at all of the dahlia gardens soil tests that I've seen, you can see that the levels of phosphorus, magnesium, and potassium in our yards is way off the charts. These little arrows down on the left, representing the three different colors and the three different minerals, those are what are considered optimum. So we are off the charts in that. Any soil scientist that looks at this says, I don't know how you can grow anything. But obviously we're proving them wrong every day. And what they will point to is things like too much phosphorus in your soil is going to interfere with nitrogen absorption, which I don't think we are going on. But it's important to realize that when you get a soil test and you see levels like that in your soil, that means you don't need to keep adding that stuff to your soil. It's already there. Focus on adding nitrogen alone. The other is there. Why it'll just keep getting higher and higher and higher, and at some point probably it does become detrimental. All right, so very quickly, here's a summary of sort of the ideals if you're looking at a soil sample test. Sandy loam, that is the type of soil that's ideal. Again, if you have a clay soil and the whole process of trying to amend it seems like it'll take forever, I refer you to the last bulletin in which people are now starting to grow dahlias in bags of dirt above the ground. And so now you can pick your soil and get the absolutely perfect soil. And as you will see, it works very well. The cation exchange capacity, the higher the better. The percent base saturation, this is the thing you should look at to try to make sure that your calcium, magnesium, and potassium are following into that desired range. The soil acidity, again, 6.5 to 7.5, I think you're going to be fine, particularly if you have a very high organic content of your soil. The soil organic matter tends to neutralize the acidity in your soil. And then the macronutrients, the phosphorus, magnesium, potassium, again, if they're off the charts, experiment. You'll see that, you really don't need to add any more. It's already there. All right, oftentimes the soil test is going to give you recommendations. Mostly it's a recommendation for changing the pH, the amount of nitrogen to put in. Sometimes if they notice, for example, that your phosphate levels are very low, it's going to give you a recommendation for how much phosphate to add. So that's something, you know, that they will help you out with. And I'll just sort of end with this. Based on all of this, the ideal fertilizer for dahlias is one that maintains a ratio of 3 to 1 to 4. So this again is the nitrogen to phosphorus to potassium. And that's going to give you, usually the 18:6:12 is the one you should look for, and that basically provides those nutrients in the ratio that dahlias prefer. Now, if you're like most of us and the charts, the levels of those phosphorus and magnesium and potassium are off the charts, there is a product called Nitrofoam. It's a slow release nitrogen only. And I've used it for years and it works extremely well. I basically accepted the fact, and I'm not going to argue with you, it was hard to do the first year to just switch to a nitrogen only and not add any more phosphorus and potassium, but it made absolutely no difference. And if you look at the soil tests, you'll see that those numbers do start to come down because the plants are pulling it out of the soil and using it, and you're just not continuing to add it. But if the levels, and it'd be unusual if your levels of those compounds are normal, then using that 3 to 1 to 4 will maintain that level in your soil. So that's what I had to say, and I guess we have time for a few questions if you have any, but that's the end of my presentation. Yep.
You mentioned to increase your organic matter by adding compost or by adding mulch. What about compost?
Yeah, same. I shouldn't have used those interchangeably, but compost is the ideal because that's going to be partially decomposed organic matter.
Okay, thank you.
Mulch is basically, can be leaves or just straw or just the coarse stuff. Yeah.
Question, where to take the soil sample?
Most, like in Michigan, Michigan State, I mean, usually it's universities that offer it, but there are also a lot of private corporations that test farmers' soil. And I think the ADS website has a list of sites where you can submit soil samples.
Yeah, I mean in your garden.
Oh, in your garden, what you should do, and I think, I'm going to do a few experiments, but in your garden you should basically take numerous samples to try to get a random picture of your soil. So what they suggest is, digging an 8-inch hole and taking a teaspoon from each hole and mixing it all together. You're only going to need to submit a half a cup of soil. So obviously you don't want to go out and take it from one part of your garden.
And mixing it all together.
And then mixing it all together. What I'm a little bit, what I wonder, for example, is we usually take soil samples long after we've dug all of our tubers out of the ground. And probably most of us go down the middle of a particular plot. We're not sampling right where that root zone is. And it would be interesting to take soil samples directly where the tubers are versus away from the tubers to see whether that high level of those compounds is actually away from where we typically are planting.
pH for fungal growth, what would be ideal?
I don't know exactly, but I think it's about the same as microbes.
Okay.
The advantage of fungal growth is that it can break down more of the more coarse carbon material that microbes can't. And so I'm not exactly sure what the pH would be, but it's...
Yeah, I have a question.
Yep.
We have clay soil. Every spring we add compost and manure, and we do a pretty heavy rototill. But now you're saying that we're destroying the beneficial fungi. How do we balance that with the need to continue?
Well, I think that when you're trying to amend the soil, that's going to be necessary in order to incorporate it into the soil.
Okay.
So you're going to need to do that until you get the soil to a level where that you're satisfied. It's not going to once you've got the good mix, then you don't really need to till anymore. But
Okay, great.
to start out, you definitely need to.
Is when you put compost in your garden, what should the ratio be from nitrogen to carbon?
Ideally, you want a ratio that's between 10 and 20, less than 20. You want a low number because you want more nitrogen relative to the carbon. And that's because the microbes are going to need the nitrogen in order to break down the carbon. And so this is why when you put raw mulch down, you want some dry brown material, like straw, but then you also want some green material, which is going to provide more of the nitrogen, like grass clippings or, clover or some of the other things. But that's the ratio of what's called nitrogen to carbon. And that's something that in that new Haney soil test, they will give you that number. And then that way you can see exactly what it is.
Then you add that to your soil test, so they charge you extra for that.
Yeah, and unfortunately it's more expensive than a regular soil test. And the one that I've been using that I'm aware of that does it is there's a corporation in Michigan called Dairy Dew that they make a lot of compost and soil amendments, and they send it out, but you can arrange through them to have that test.
I hear that avoiding oak leaves, if you're going to use leaves, is that true?
I mean, oak leaves are very acidic, and they're high in phenols, and they are very slow to break down. So ideally, you're going from year to year, you want to put in things that are going to break down more readily. And that's why if you choose leaves like ash or maple, beech, those tend to break down much more quickly, and they don't have as much of the phenolic and acidic compounds.
Is there something you can add to the water to act as a catalyst to help it break down faster?
There are products that are designed that farmers use. For example, corn farmers, they go through and cut the form, and then the corn, and then they have all of those stubs. There are products that are designed that have a very high fungal population, which help break that stuff down much faster. All right, I guess we got ready for the next talk.